Hello, good morning to all the person connected with this new edition of the ESCMID webinar, and it's a great pleasure to have you here in this afternoon. And the topic we will stress today is a very hot topic because it's a vaccination, and we have for this topic two outstanding speakers. The first is Marco Cavaleri from EMA. Marco is the responsible for EMA for vaccines, so he's the right person in the right moment to explain us all the problems, all the story of the vaccine for COVID, for SARS-CoV-2. And the second speaker is Khitam Muhsen from Tel Aviv, Israel, and she will share with us all the experience of Israel about the COVID-19 vaccination. So, okay, it's a great pleasure for me and an honor. And thanks so much, Marco Cavalieri, to be here in this afternoon. And Marco will have a talk about the SARS-CoV-2 vaccines, the EMA perspective. So, please, Marco.
Hello to all of you. I hope you can see the screen.
If you can put a full screen, it should be perfect. Perfect, Marco. Thank you.
Okay. Okay. So indeed, as Maurizio said, in my presentation, I will go through briefly where we are with vaccines for COVID-19 and very much focusing on the one that have been approved. But let me start one step before, just to mention the variety of vaccines that are under development for SARS-CoV-2. You know, there have been more than 250 candidates that have entered preclinical development. And as you know, the majority of these vaccines are focusing for a number of good reasons to the spike protein, and some of them even just focusing on the so-called RBD, so the receptor binding domain within the spike protein, even if I have to say the majority of the candidates for various reasons have decided to go with the whole spike protein. And indeed, most of the vaccines that we have now and that are under development as we speak are using the sequence of the so-called Wuhan strain, so the ancestral strain that was at the basis of this pandemic. While, as I will tell you more later on, now we have to face what to do with variant strains that have been emerging recently, and there are reasons for concern, particularly in terms of antigenicity. And in terms of the platform technology that have been employed, we've seen a variety of different ones that have been considered. So aside from the more traditional one based on inactivated virus, whole inactivated virus, or recombinant protein, we have seen particularly the emerging of the RNA technology as a technology that can deliver vaccines really rapidly and more rapidly than other technologies. And on top of this, with amazing results, as you all have seen from the clinical trials that have been conducted so far. But beside these that are under I here on the slide, also H is an important technology under consideration, and this is the use of viral vector vaccine, particularly the replication incompetent one, and the adenovirus has been among those that have been preferred, that have been brought forward for a number of constructs that have demonstrated already efficacy in clinical trials, and some have been already approved. As you see, there are many others. We will see where they're going. We are working with a number of developers looking into all these different technologies, and we will be very happy to see whether any of these or maybe all of these will be able at the end of the day to be successful. But let's focus on what we approve, at least in Europe, as EMA. And I will start indeed with the two messenger RNA vaccines. These were the first one to be approved. And just to mention that indeed the European approval is a real approval, is not an emergency use authorization as has been done in other parts of the world. Even if this approval came as a conditional marketing authorization, so which means that some of the data that normally we would like to see pre-approval will be generated post-approval and will be submitted later on. As you know, these two vaccines, essentially the one from Pfizer BioNTech called Comirnaty and the one from Moderna, they utilize two doses in order to achieve a good level of protection. given after with an interval of 3 weeks in the case of Pfizer-BioNTech and 4 weeks for the Moderna one. And as you know, the results of the large clinical trials, placebo-controlled trials, were showing a level of efficacy around 94–95%. Here it's important to stress that we are talking about protection from disease of any severity, and in the vast majority of the cases that were collected in these event-driven clinical trials were essentially mild disease. So we are talking mainly about protection from mild disease, and you know also the difference in terms of storage condition with particularly the Pfizer-BioNTech that has to be kept at -70, even if you may have heard that there are new emerging data around stability that will show that it's possible to maintain also longer term this vaccine at kind of higher temperatures, while for the Moderna one is about -20 degrees, but also for limited amount of time can be kept also in the fridge. Now, the other important point that I wanted to mention to you is that it's pretty clear, as you see here on this plot, on this Kaplan-Meier, that the efficacy was evident after the first dose and roughly after 14 days from the first dose. In the graph, you see what is happening for the Pfizer-BioNTech vaccine, but similar considerations would apply also for the Moderna one, which shows that even after a first dose, you start to see efficacy, which get consolidated after the second dose. And let's not forget that the second dose was given almost all of the cases in the clinical trials with a window that was not behind 6 weeks from the first dose. And what is important to stress is that the reason why the companies went with the 2 doses is that indeed after a single dose, as you see here on these slides in terms of neutralizing titers, after first and second dose for the Pfizer-BioNTech vaccine, you see clearly that at day 21, so just before the second dose, the level of neutralizing antibodies is extremely low. With all the doses tested, and here was a dose finding study. Just to remind you that 30 microgram is the dose that was selected then for the large clinical trials and is the dose that is approved. But even there, you see that the amount of neutralizing antibodies are really low and really close to the limit of quantification, while you get a dramatic boost of these antibodies once you receive the second dose. Even after 7 days or within 7 days, you see really a tremendous boosting of this adaptive response with really high level of neutralizing antibodies, even superior to what we see with the panel of convalescent plasma. So this just to stress why 2 doses are felt needed and why it's so important to take into account the impact on the immune response after a single dose versus 2 doses. And something similar was also seen with the Moderna vaccine, which indeed use pretty much the same technology, even if there are some subtle differences in the lipid use and the amount of messenger RNA incorporated. And also here you can clearly see that just before the second dose, including in the elderly, the amount of neutralizing antibodies are really low and even below the threshold of quantification. So really showing that you need 2 doses in order to boost a very good humoral response with really high level of neutralizing antibodies. And that's why it's so important to try to stick as much as possible with this posology until there is new evidence emerging that would show us what is the level of efficacy with a single dose more longer term. But also in light of the emergency of the variants, it's important not to forget the value that high level of neutralizing antibodies can have. Now, very rapidly moving into the safety, as you know, essentially reactogenicity has been the main area of focus. So overall, these vaccines have turned out to be pretty safe with really mild or moderate reactogenicity that has been shown both in terms of local and systemic signs and symptoms. Here you see the data after the first and second dose for the Pfizer-BioNTech vaccine, in which you clearly see that there is an increase of the reactogenicity after a second dose. Here is what is in the slides is you see systemic reactogenicity and fatigue, headache, chills and pain being the main one seen with this vaccine, which is very interesting because a similar pattern has been seen with Moderna, but with The AstraZeneca vaccine, most of the reactogenicity occur after the first dose, while after the second dose seems to be much more attenuated. And overall, in terms of risk, this slide summarizes what has been included in the risk management plan for the messenger RNA vaccine. This is specifically for Moderna vaccine, but is exactly the same of what we have stated also for the Pfizer-BioNTech vaccine. The only important identified risk so far is anaphylaxis, and this is something that emerged really immediately with the start of the vaccination campaign, particularly associated with individuals with an history of serious allergies and previous history of anaphylaxis, but also in few cases without that. That's why it's so important that there is close monitoring of vaccinees at least for 15 minutes or even half an hour after receiving the dose of vaccine to make sure that there is no emergent risk of anaphylaxis or anaphylactoid reaction. Another important potential risk that is still present is around the possibility of having enhancement of disease, even if this seems to be more theoretical than real. It was a big concern at the beginning based on what we know from preclinical models of other coronaviruses such as MERS and SARS. But so far, we have not seen any clear signal that would point to any risk of vaccine-associated enhanced disease with this vaccine in preclinical models or in clinical trials. Of course, it will be important to follow once the antibodies will decay to see what is happening, but again, nothing to be worried for the time being. And then, of course, there are a number of missing information, as often happens after the initial authorization of vaccines, starting from the use in pregnancy, which anyway is allowed if the benefit is considered to outweigh the risk, and considering the COVID-19 can be quite deleterious for pregnant women. As stated, the long-term safety is a bit of an unknown at this stage, but we know that most of the adverse drug reaction to vaccination occur within the first 2 months after vaccination, but still is important to follow up and monitor more closely what happens in the long term. The use in immunocompromised subjects, we don't have evidence as these subjects were essentially excluded from the pivotal clinical trials, and it will be important to follow on, particularly with dedicated clinical trials looking at the immune response, but also into effectiveness studies. Interaction with other vaccines, such as influenza vaccines, will be important to collect, and other evidence in the context of using frail subjects or subjects with autoimmune or inflammatory disorders will also be important. Now, let's move to the third vaccine that has been authorized in the European Union, which is the one from AstraZeneca, which uses indeed a viral vector, which is the chimpanzee adenovirus 1. And this is what is depicted in this slide. Essentially, what they included into this adenovirus has been the linearized form of the spike protein, so not in a perfusion conformation. But this allowed them to move faster into clinical trials later on. And as you know, initially the idea was to use a single dose of this vaccine, and this was based on the experience also with animal models in MERS. But then it was soon realized that a second dose would increase the level of neutralizing antibodies, and that's why there was a decision to give a second dose of this vaccine at least 4 weeks after the first one. The point, as you know, is that some of the large clinical trials already started, particularly one in the UK, already initiated with a single dose, and therefore there was a catch-up vaccination for the second dose during the clinical trials, which is what also complicated the interpretation of the clinical trials coming up, particularly from the UK, but to a certain degree also the study done in Brazil. And then another important point around immunogenicity is that, as you know, there are not many clinical data in terms of protection from COVID-19 in the elderly, and more in general above the age of 55, because most of the participants in the UK and Brazilian study were below the age of 55, or elderly were included later on. So there is not really evidence so far that would show exactly what is the vaccine efficacy of the AstraZeneca vaccine in this context. But nevertheless, we looked at the immunogenicity in the elderly of this vaccine, and you see here on the left at the bottom what happens after a second dose of the AstraZeneca vaccine in elderly above 65 years of age, in which you see really a good amount of neutralizing antibodies being elicited, which at the end of the day, considering that in the elderly, the interval between the two doses was kept within 4 to 6 weeks, you see is not different than what you see on the right in a more generalized population of adults, in which you see that when the interval is kept below 6 weeks, then the level of GMTs in terms of neutralizing antibodies is not at all different than what we see in the elderly. And what you might appreciate here is that there is a clear signal that the larger the interval it is between the two doses, the better will be the immune response after the booster, which we don't really know exactly what is driving this. It might well be that immunity against the vector is playing a role, but there are other elements in terms of maturation of the immune response that could play a role here. But the bottom line is that if you wait a bit longer, you will have a better booster response. And at the same time, the immune response in the elderly is not significantly different than in adults. And that is the reason why the EMA approved also this vaccine in the elderly, despite we don't have yet a clear-cut estimate in terms of vaccine efficacy. And this is essentially the vaccine efficacy that has been demonstrated in the pooled analysis of the UK and Brazilian study using the interval that has been claimed by AstraZeneca. So the interval between the two doses that have received approval, which is an interval of 4 to 12 weeks between the two doses. And taking into account this population in the pooled analysis from the two trials, what comes up is an efficacy that is close to 60%. If you look at the overall efficacy in the study, so taking into account also subjects that received the second dose much later than 12 weeks, the efficacy goes up to 62.6%, so not a big difference. And indeed, the interval here would go up to 23 weeks after the first dose. And what also is important that also with the AstraZeneca vaccine, we are seeing protection already emerging after a single shot. And after approximately 14 days from the single dose, you see that the two curves between the control and the vaccinated subject with the AstraZeneca vaccine, really they are separating, and really there is efficacy demonstrated already there. One of the problems that we had was really to have a clear quantification of the level of efficacy after the first dose up to a period of 12 weeks. And that was very complicated because of the fact that many individuals in the trials received the second dose at different intervals. And also there was a differential outcome in the study between Brazil and UK, which made very difficult to reconcile the data and to come up really with a clear-cut estimate of the vaccine efficacy after a first dose and for a period of 12 weeks. But the other important point, even if the data are extremely limited, is that what we're seeing is that it appears to be a good protection from hospitalization and possibly from severe disease. The numbers are very small, so we are talking about 0 cases in those that received the AstraZeneca vaccine versus 8 cases of hospitalization following 2 doses as per the approved use of the vaccine. But also importantly, we have seen that even after a first dose, there were 0 cases of hospitalization against 14 in the control group. And in the control group, there was also 1 fatality. So even if the data are limited, still the signal is pretty clear that this vaccine seems to be able to protect from progression of disease, hospitalization, and possibly severe disease. Of course, we will need more data that will come from real-world evidence and other clinical trials that are ongoing, but these data are important and could support the use of this vaccine, including for groups that are more at risk of progressing to severe disease. What about the safety of the SHADOX1 vaccine? Essentially, also here, what we've been seeing was a reactogenicity, which was mild moderate, particularly after the first dose, then more than the second one, differently from the messenger RNA. And malaise and other systemic events like headaches and fatigue were reported quite frequently. As said, more were mild or moderate and self-limiting, and the duration is no more than 3 days. even after the second dose, but almost 10% of subjects experienced grade 3 systemic adverse event, which essentially were related to fever and other systemic reactogenicity events. In terms of serious adverse event, there was not much reported in this study, so overall it looked quite clean so far, the safety database from this perspective. As you know, there was a case of transverse myelitis that attracted a lot of attention in the clinical trial, but there were no other cases, and so far there is nothing else that would be of concern in this regard, even if all the neuroinflammatory events are part of the adverse event of special interest and will be followed up quite closely in the post-approval phase to make sure that we will capture any signal that will be emerging in this sense. Now, a few words around the variant. And as you know, the variants have been emerging in different parts of the world. The so-called Kent variant that emerged in the UK, so the B117, now is spreading all over Europe and in other parts of the world. But what you see here on these slides are data in terms of cross-neutralization from sera derived from vaccinees that received the messenger RNA vaccine. So on the left is the Moderna one, and on the right are data with the Pfizer-BioNTech. So what you see on the left with the Moderna on the top is that essentially there is no impact in terms of cross-neutralization of the B117 variant on the ability of the sera to cross-neutralize also this SARS-CoV-2 mutant. And this has been confirmed also with Pfizer-BioNTech. A bit more complex is the situation with the so-called South African variant, which is the one at the bottom here on the left, the B1351, that, as you know, has 2 additional mutations in the receptor binding domain and a total of 10 mutations in the spike protein. And what you see here is that indeed this has an impact on the cross-neutralization potential of this vaccine. And the same can be said for the Pfizer-BioNTech that you see here on the right. So really the ability of the sera generated by this vaccine to cross-neutralize this variant seems to be inferior. But nevertheless, we really don't know, since we don't have a culture protection, what exactly means in terms of protection in the field. So the question is still open to what extent this reduction in cross-neutralization will indeed impact the level of protection from this vaccine. And not only in terms of protection from mild disease or maybe asymptomatic infection, for which we don't know yet, by the way, with this vaccine, but it will be very important to understand what is happening in terms of severe disease. And as you know, something that is more of a concern are the data that have been emerging from clinical trials conducted in South Africa with the SHADOX1 vaccine, so the AstraZeneca vaccine, in which they showed that there was really not a significant effect in terms of protection from mild disease of this vaccine. In the context of an epidemiology that showed the B1351 variant being the main strain circulating in South Africa. So this is of concern, because it would mean that, at least in terms of mild disease, this vaccine would lose protection. We have seen also data from Novavax and Johnson & Johnson that would show also some reduction in the level of protection from clinical trials conducted in South Africa as well, even if maybe not as marked as this one. But again, it will be very important to understand if this reduction in protection from mild disease will also translate in reduction in protection from severe disease. And we don't know that. And it will be extremely important and valuable to get this data, some data in this sense, because it might give us a bit of a different picture and something more positive with respect to protection from hospitalization and severe disease. To conclude, there are different technologies that are under investigation. 2 messenger RNA vaccines have been approved that require 2 doses with very high efficacy in protecting from COVID-19 of any severity, including in the elderly, and overall very much benign safety profile, with reactogenicity essentially being the major problem so far, and anaphylaxis in few cases. The interval between the 2 doses should be maintained as much as possible in line with the SMPC, as indeed there are too many uncertainties at this point in time with respect to what a single dose can do, particularly in the long run, and taking into account the low level of neutralizing antibodies. The Shadox-1 vaccine, which also has been approved in the European Union, showed efficacy around 60% for protection from disease, from COVID-19 of any severity after a second dose and with an interval of 4 to 12 weeks. But efficacy was shown also after the first dose and seems to be suitable for giving sufficient protection up to 12 weeks. This vaccine is generally safe with more reactogenicity in adults than in the elderly. And indeed, we concluded on a positive benefit risk also in the elderly, taking into account the safety profile and the level of immunogenicity achieved in this population as well. All the vaccines studied included subjects with comorbidities such as asthmatic, COPD, cardiovascular, and diabetes and other comorbidities, but not immunocompromised patients that were excluded from this trial. So it would be very important to fill this gap and generate data in the post-approval phase in this population. And as said, in light of the emerging variants that might show escape immunity, the EMA, similarly to other agencies like FDA, just issued their own guidance. We are going to also publish very soon a guidance document that will describe what could be a streamlined regulatory pathway that would allow rapid approval of new vaccines that include some of the variants of major interest. And then lastly, very important to look into the post-approval phase, not only with randomized controlled trial, but also with studies looking at effectiveness and safety in large population in order to have a very good characterization of the benefit risk of all these vaccines. So thank you very much.
Thank you so much, Marco, for this very comprehensive and clear, very clear presentation. I think is very, very good for all us. And okay, we have some questions from the audience. The first from Victoria is, how come after the first dose of the vaccine there is a decrease in clinical disease, but not in levels of neutralizing antibodies?
Yeah, so essentially, what we're seeing is, and I don't know which vaccine you were referring explicitly, but you know, as I showed to you, it happens that was a bit surprising, I have to say, that we saw good protection after a single dose of the messenger RNA. Again, we are talking about very limited intervals because after 3-4 weeks max, they received the second dose. But the efficacy up to that point in time, it looked pretty good and more than what we could have expected. And now we don't know exactly what is driving this protection. Because indeed the level of neutralizing antibodies is really low. And so there are probably other elements here that could play a role. The T-cell responses are important, but they were not very well characterized. And we cannot also exclude that the activation of the innate immunity could play a role as well. So it will be extremely important to do some more work on the immunological side in order to try to get a better insight and understanding of what is driving this protection, and also importantly, whether these could be long-lived behind, you know, the small interval that was studied here.
Thank you. In fact, one was my question about T cells and vaccines, but we have several questions from the audience, so it's better to reply to the audience questions. So the other one is the relationships between pre-infection with SARS-CoV-2 and vaccine. So there is some importance for the presence of antibodies from a previous infection and vaccination. And of course, it is necessary to modify the vaccine schedule for person who did COVID disease.
This is a very important question, and I have to say that subjects with the history of COVID-19 were excluded from the clinical trials, but nevertheless, there were a small group of subjects that were seropositive at baseline. So probably they encountered the virus, but they were not aware that they were infected, or at least this was not reported. And what we are seeing that this small group of seropositive, overall, the vaccine was safe, so there were no problem to administer the vaccine to these people. But in the few cases in which the immune response was measured, really showed that the level of antibodies really goes up dramatically, and indeed it might be fair to think that a single shot could be sufficient for people with a history of COVID-19. So we need to collect more information in this regard, but I believe that overall it could be a reasonable strategy to think about giving a single shot of this vaccine for subject with a history of COVID-19. One of the problems, you know, is that this population is extremely heterogeneous and the level of immune response can vary dramatically. So I cannot exclude there might be some subjects that might still deserve to get also the second dose. But on average, at this point in time, it might be reasonable to go with a single shot.
Perfect. In my very small experience, I agree with you, because we had some healthcare workers with disease and with major with some adverse effect during the first shot, so very high level of antibodies. And finally, last question, then we can go ahead. The immune response for how long post second dose vaccine? So how long, of course, this is difficult to know because we are in the progress of our experience, but in your opinion, how long can be the protection?
Yeah, it's an important question. And so far we've seen data only from Moderna after 3 months that showed that the level of antibodies are still maintained quite decently. So it looks like probably if there is not a major drop, which we don't know yet. So we need really to look into the data from the studies that are ongoing with those that received the vaccine a year ago or something like that. But it might well be that you will still maintain good level of antibodies up to 1 year. But again, we need to have the proof of that. So it's very difficult to say at this stage and is a bit speculative.
And when the full official approval by EMA?
Yeah, that's it can well be that it will come in 2021. We will need to wait to receive a number of data on the manufacturing side, and we are already receiving quite a lot of them. Plus, we are look forward receiving the final study report from these clinical trials. As you know, we were hoping that this placebo-controlled trial could still go on for a while so that we could have comparative data for a year or more. But clearly, with the vaccination campaign ongoing, it would not be ethical to maintain particularly subjects that are in risk group into these clinical trials if they had received placebo. So we have to accept that there will be crossover. Nevertheless, it will be important to get this information and to get longer-term data on the safety of this vaccine from these clinical trials.
Okay. Thank you so much, Marco. Really appreciated. And we can move to the next, a final presentation by Khitam Muhsen about the nationwide SARS-CoV-2 vaccination strategy, the case of Israel. Of course, the data from Khitam will be very important for us. Thank you once again for accepting our invitation. Please, Khitam.
Thank you very much for the invitation. I'm very happy to share with you our experience from Israel. Can you see the slides now? Okay, so just a minute. Can you see the slides? Is that okay now?
We see.
Yeah.
We see your slide.
Good. So I will start. So thank you for the invitation. It's really nice to be with you this evening and to hear the impressive presentation of Marco and the good, excellent questions. I enjoyed it very much. I will provide information about the epidemiology of COVID-19 in Israel in order to better understand the context of COVID-19 vaccines in the country. And the second part, I will focus on COVID-19 vaccines in Israel. So far, this is the map of the world with the COVID-19 cases. More than 110 million cases all over the world, including 2.5 million fatal cases. So it is really very difficult epidemic. And our region, the European regions and the Americas are the most affected in terms of number of cases as well as a number of fatal cases. So a heavy burden of COVID-19 in our region in Europe. Israel is a relatively small country, and this might explain the success of the COVID-19 vaccination. We have 9 million people. The population includes 9 million people. About the majority are Jewish, and we have an Arab minority that comprises 21% of the population. And we have also another subpopulation within the Jewish population, which is the ultra-Orthodox Jewish. It's a religious community. And it is important to know about these communities because the epidemiology was different in this population compared to the rest of the countries, and also they differ in terms of socioeconomic status and the COVID-19 vaccination. So I will explain about this, but it is important to know about these subgroups in the population. A few words about the healthcare system in Israel. It is similar to many European countries. We have the Ministry of Health, which is the main regulator. We have universal health care. All citizens receive a basket of health services, mostly provided by the health maintenance organizations, free of charge. This includes primary care, outpatient visits, hospitalizations, and we have clinics and hospitals that are spread all over the country. So access to care is relatively high in Israel. This is important also because it affected the testing for COVID-19 and also the vaccination campaign success. If we look at the PCR testing in Israel for SARS-CoV-2, initially testing was limited to people with the symptoms and to people with epidemiological link to confirmed cases. But with time, testing was offered to almost everybody, especially to contacts of confirmed cases, even though they didn't have symptoms. And the reason for that is to detect asymptomatic individuals that can transmit the disease. And we see that the capacity of the labs increased and testing increased substantially with time. So this is the number of cases. Overall, the cumulative number of cases is about 700 cases all over the country. PCR confirmed cases. We see 3 waves in March, April. This is the first very small wave. A second wave in September, and the third wave last December and during January 2020. And the prevention and control measures are similar to the rest of Europe, or travel restriction, contact tracing, quarantine, lockdowns. And we see when we lift the lockdown, the incidence increases. A second lockdown, the incidence decreases. And it increased again with the reopening following the second lockdown. In December 19, we started the vaccination, and in last December 27, we went into a third lockdown because of the high incidence of the disease. Each wave was followed also by increasing the severe cases and in the fatal cases of COVID-19 in the population. The incidence rate, this is also important to vaccination. We see the highest incidence in the young age groups, and the incidence decreased in older ages. However, with fatality similar to other European countries and other countries in the world, the mortality rates are highest in people aged 60 years and older. We see the differences in the disease incidence, with highest in ultra-Orthodox Jewish population and the Arab population, and also the incidence decreases with improved socioeconomic status. So now I will move to vaccination that we started on December 19. But before I go into the vaccination campaign, I want to tell you a few words about the strategy with COVID-19 vaccines. At the early stages of the pandemic, actually, the government and the Ministry of Health made contact with several vaccine companies, and there were several contracts in order to buy option, and with Pfizer, Moderna, AstraZeneca, and the Russian company Sputnik 5. And I think this strategy is good because at the early stages, we didn't know which vaccine will make it till the end, till the clinical, the phase 3 clinical development. So this was the rationale. And in addition, we have a local vaccine which is under development. The Pre-Life vaccine, it is a recombinant vesicular stomatitis virus is used as the vector, and it expresses the S antigen, as mentioned in the talk of Marco. And this vaccine currently is in phase 1 and 2 clinical trials that were already completed. So we began vaccinating the population with the Pfizer vaccine in December 19 with 2 doses given 20 days apart. The vaccine is given free of charge, voluntary, no obligation by law. Vaccination is given in HMO clinics and also in mobile centers to reach to remote areas and to isolated the population. Also, vaccination is done in nursing homes. Initially, the groups that were at the first priorities were healthcare workers, people aged 60 years and above, and people with the underlying diseases. And now the vaccine is open to everybody above the age of 16 years and over. And a few days ago, the Ministry of Health recommended the vaccination to people who recovered from COVID-19 with only 1 dose. So people who have recovered from COVID-19 should receive only 1 dose. This is the new policy. And we see Israel is ranked first as in the number of COVID-19 vaccines per capita, followed by the United Arab Emirates. So far, about 47% of the population received the first vaccine dose, and about 1/3 of the population received the second vaccine dose. This is the cumulative vaccination percent. The first dose in blue and in orange is the second dose. So it is really a great success to see this huge coverage and uptake. And I think people were really happy that this vaccine became available. The campaign is called Back to Life. We are all want our life back. So the uptake so far is very, very good. Now, if we look at the vaccine uptake by age groups, we see excellent uptake exceeding 80% in people aged 60 years and over. Almost everybody was vaccinated above the age of 60 years. We have a problem with the young age groups. We need to invest more effort convincing the young people to come and vaccinate, to get the vaccine. But I think this will improve with time. But overall, excellent vaccine uptake in the main risky groups for severe disease and mortality. We see also social disparities in the vaccine uptake. The vaccine uptake is lower in the low socioeconomic status community. It increases with improved socioeconomic status communities. And also it is lower in the ultra-Orthodox and the Arab population compared to the general Jewish population. So much of explanations and the education is done in these communities to improve the uptake, including socially tailored interventions and educational interventions in order to encourage people to receive the vaccine. Vaccination also is being completed for a non-Israeli who live in Israel. This is last week from Tel Aviv. People who do not have Israeli citizenship also receive the vaccine free of charge, of course. And this is me receiving the first dose of the vaccine, waiting for the second dose and receiving the second dose. I survived, it was okay. So with regard to monitoring the side effects or the adverse events following vaccination, I present here data of the Ministry of Health. It is this monitoring is done through passive surveillance. The data here are related to the first 3 million first doses and about 1.5 billion second dose. Overall, the most side effects or adverse events were mild, transient. This included local and systemic adverse events, mostly local adverse events that included pain at the site of the injection, and general side effects that included mainly fatigue, extreme fatigue, headache, and fever. Other side effects or adverse events were relatively very rare. So we can say up till now that the adverse events are mild and transient, including both local and systemic. And it is interesting that we see that the young age groups, 30 to 49, reported more adverse events than older age groups. And also we see that women more than men reported adverse events following vaccination. And if we look at the early evidence of vaccine effectiveness, this is the real world early evidence. In this study, we used the data from one of the largest HMOs in Israel. We used data of about 500,000 vaccinees who received the first vaccine dose, and we followed only the vaccinated individuals. We had only vaccinated individuals, and we see that the incidence was lower during the after the day 12 following vaccination compared to the early period, days 1 to 12 after vaccination with the first dose. we were able to calculate the vaccine effectiveness of 50% after the first dose, which was evident up to 24 days after the vaccine, after the first injection. We are currently working on this study to refine the analysis and also to include the analysis of the second dose. Data reported by the Ministry of Health showed the effectiveness after 2 doses, 7 days after 2 doses. It exceeded the 90%, 95% against COVID-19 hospitalizations. And the effectiveness was also very high, exceeding 95% after 2 weeks after completing the second dose. So really encouraging results. Really, we can see the light at the end of the tunnel. Another study from Israel conducted in healthcare workers that was just published a few days ago showed effectiveness of almost 50% during the first 14 days after vaccination with the first dose, and they have a higher effectiveness estimates of about 75–85% 2 weeks to 4 weeks after the first dose. So it is really encouraging and seems very nice. This is the incidence of COVID-19 in individuals aged 60 years and older. The yellow line and the blue curves are the vaccine uptake, and we see towards the beginning of January a decline in the incidence. Also in COVID-19 hospitalizations in this age group. We just lifted the lockdown, started to lift lockdown lifting here. So it is important to continue these studies and the analysis and to see whether this effect is related only to the vaccine or also to other control measures such as the lockdown. But it looks promising. But despite this success and very exciting vaccination campaign, we still have challenges. We have to increase the vaccine uptake in the young age groups, in minority groups, in the Arab and the ultra-Orthodox communities. And we do that in using positive incentives to there's another way to enforce vaccination by law, but still this is in debate in Israel. So we prefer to use positive incentives. Also, the new variants of SARS-CoV-2 were introduced to Israel. Over the past few weeks, a high percentage of the circulating COVID-19 SARS-CoV-2 were actually attributed to the UK variant. The South Africa variant was also introduced to Israel, but it is in a very low proportion still. Again, as Marco mentioned, we still do not know the duration of protection, so we'll wait and see. And without a pediatric vaccine, we expect that the circulation of SARS-CoV-2 will continue in the population, in the young age group. And we need to see how the vaccines will work in immunocompromised patients in real world actually. And another word that we have is fighting the fake news, because many of the concerns and people do not want to take the vaccine because of lots of fake news on social media. So lots to be done in this area as well. I think with that, I would like to thank my colleagues who contributed substantially to this work, Professor Danny Cohen, Dr. Lena Lapidot, Jonathan Amir, Dr. Wasif Naaman, Sophie Gorin, and Sarit Perman. Also my colleagues from Maccabi, Professor Gabi Khudik and Lilach Tene. And with that, I thank you, and I'm happy to take any questions.
Thank you so much, Khitam. Very nice presentation, very encouraging data. Thank you, because this is very important. So as you can see from the chat, we have some questions for you. The first is, do you have some data on population level seroprevalence before introducing the vaccination, or this was not important for you?
Yeah, so we have data. The largest national sero-epidemiological study was actually conducted around September 2020, and it showed about 5% seroprevalence in the general population. So since then there is no additional, there was no new sero-epidemiological studies. We did some studies in healthcare workers, in multi-center studies, and we found about 8 to 9% during September till November. So these were the estimates before starting vaccination.
Thank you. Another interesting question is about ultra-Orthodox Jewish population in London, because it seems that it was reported to have tried to attain an herd immunity by natural infection with low deaths. You have information about this? Which possible reason for this situation?
Yeah, so the ultra-Orthodox Jewish community is characterized by large families and living in crowded conditions. So the transmission of the virus is efficient in these households compared to the general population. The incidence was really high in the ultra-Orthodox Jewish community in Israel, and it reached about there are estimates of about 10% to 15% of the population was infected in these communities. But this is very far from the herd immunity level that it is needed to block the transmission of the virus. So
yes,
vaccine will do the work.
Yes, exactly. Okay, about the question about the variants, you are already exposed in your presentation, if I remember well, because you have South African variant, but it's not clear if there are some problems with vaccination. So what about the screening, after the second dose of the vaccine? In other words, do you use a rapid antigen test or other molecular tests to screen population? And you plan to continue this approach also after the second vaccination or not?
So I think the idea is to reduce testing for people who get the second vaccine dose. So this is the idea. I think testing will not be requested, and people who get actually the 2 doses of the vaccine actually will receive what is called the green passport. What is the green passport? These are the incentives. You will be allowed to go to theater, to attend the sport events, to go to the gym. So, and people will not be requested to be in quarantine if they are vaccinated actually.
About the type of vaccine you are using only Pfizer, or you are using also other vaccines such as Moderna, AstraZeneca, Sputnik?
Yeah, so currently we are using only Pfizer. We received a few hundred of doses of Moderna. Yeah, after making so many contracts. So, but I think the Pfizer vaccines or the available doses currently will be consumed, and we will switch at some point to Moderna.
Okay, you are receiving a lot of questions, Khitam. Do you organize some matrix to notify all adverse effects, including that case in elderly or in other immunosuppressed or fragile patient?
Yeah, so because we at the HMOs we have electronic health records, so physicians are requested to report adverse events to the Ministry of Health. It is like in their computer, somebody comes with the fever, they ask the patient, is it related to the vaccine? Have you been vaccinated? And the physician has to fill in this information. So the information is collected in some way in an active manner, but it is also collected in passively. So people who have adverse events are requested to report to the Ministry of Health.
Okay.
We don't know who reports what.
Okay, final question. Your opinion about mRNA vaccine and attenuated vaccine. You are what do you think about this?
The attenuated vaccines and which one?
And mRNA vaccine, the Pfizer or Moderna.
Yeah, it seems so far that the mRNA vaccines, the data looks promising regarding possible effect and reduction of disease and infection, reduction of the risk. So, and the safety profile in the general population seems okay. It's mild, moderate, very few severe or adverse events. So I think these are okay.
Okay, so no more question. Also no more time. We finish absolutely in time. So it's a great pleasure once again to thank Marco and Khitam for this very nice lecture, and this, for me at least, very informative, very important, and very also hot, because it's absolutely in time. So thank you so much. And thank you so much to the audience for to be part of this event, and also for very active discussion. This is very nice. We received a lot of questions. Thank you so much to the ESCMID office to organize this event, in particular to William Cuddy, who is absolutely active and absolutely fine in this activity. Please not forget that also next week we'll have other two webinars on COVID, in particular for immunity, and for the treatment. And thank you so much once again, and have a nice evening. Bye, and see you.
Thank you.
Thank you so much.